So our final speaker in this session is Lisa Brenner. Professor Brenner is at the University of Colorado, Denver School of Medicine.
I think they have my slide deck up there, so I'm going to just trust that that's going to happen. And I'll start out, this is like now for something completely different, or like what's a psychologist like you doing in a place like this? And so the official title of my talk is Mental Health and the Microbiome of the Built Environment. I do work at the Department of Veterans Affairs, so everything I say today is totally Lisa, not the Department of Veterans Affairs. And I think the talk last night must have inspired all of us to think about the stories of this line of research. And I'll say what brought me to this line of research. I have been at the VA for 20 years this May, and have worked primarily at the intersection between traumatic brain injury and negative psychiatric outcomes. And many people, the Department of Veterans Affairs, many agencies, had spent a long time, including, I should say, the Department of Defense, thinking about how are TBI, traumatic brain injury, and the mental health symptoms different? Is it TBI or PTSD? And at the same time, not coming up with any particularly new interventions to help these veterans returning from combat in Iraq and Afghanistan. At a talk like this, where I think most of you probably don't spend your every day with veterans, I do like to ask how many folks you think have been deployed to the global war on terror. And so I'm not choosing that name, that's somebody else chose that name, but let's just say that's everything Iraq and Afghanistan and beyond. How many people? This is the participatory part. If you know the answer, if you've played this game before, do not play again. How many people have been deployed? 100,000. Starting bid is 100,000. This is a little bit like The Price is Right. Over or under 100,000? Over. How many? 1 million. Over or under? Over. Okay. Where? Come on. You know how to play this game. I know. You've watched Price is Right, even though you're very smart scientists. Okay, over 2.5 million. It's over 2.5 million. It's about 3 million right now. Okay, so 100,000 is usually close. I usually get starting 10,000. So this is about 3 million people to be thinking about and growing, right? We still have folks deployed. I keep hoping that someday I'm going to come up here and give a talk or somewhere and give a talk and say, you know, everybody's home from Iraq and Afghanistan, but they're not. And then we also need to think about these folks' families, their kids, and how they age. And so, can I just ask, sorry, I know there's so many technical issues. I don't see the presentation here. Should I be able to see it here? I can try to see it there and can put on my glasses, and that will help me a little bit. So the other thing that, as I became frustrated, I started to think about how can we think about this differently. And I knew that I couldn't figure this out alone, so I needed to find people to help me think about it. So to the previous comment about blogs, I was on vacation, actually walking on the beach. I don't know why I was walking on the beach reading a blog. That's me, I guess. And I found a blog by Andy Hoisington on the work that he was doing. And I cold called him and said, hey, I'm interested, this is what I'm doing. And he was nice enough to meet with me and talk to me. And we brought in some other people, and we have Kelly Stearns here. Kelly and Andy, can you just, I mean, because two of you are integral to this, they're back there. So Kelly and Andy, and Kelly keeps us all on track and is my lab manager. Thank you so much. That's very helpful. And so one of the things that Andy and I did first, this is in 2015, so just 2 years ago, was to try to create a model where we could speak each other's language, right? And so for me, it's about the individual, sick individuals, I don't like that so much, but sick individuals in sick buildings. And can we use similar models to think about those two different I guess, research topics, and start to think about how can we talk together about the people and the buildings. And so we came up with this model. Thank God this model got a decent review on MicrobeNet. And this article, it wasn't an oversell, it was just good, and that was a great day for us. And so, you know, we have been really working in two different lanes, but together in this interdisciplinary team. One is looking at the building, and so this is a lot of the work that Andy and I have been doing together with others. I do want to highlight in particular the idea that we're working on looking at the longitudinal assessment of the influence of lifestyle and the built environment on a cohort of U.S. Air Force cadets. If you don't know about this study, there will be more data coming to you. We saw a bunch of data this week. But really, this cohort of individuals, they live together. They are hypothetically the best and the brightest. It is really hard to get into the Air Force Academy. They eat the same thing. They do the same exercise. They live together. They live in rooms together. They are an awesome cohort to study, to think about how does the built environment affect people, how do people affect each other, and how does the microbiome change over time. And so I'll show you just very, very little bit of this data today, but I want you to stay tuned because I really want Andy to be able to present this work. He's done most of the work to make this happen, but I think some very exciting work about finding communities. What are other communities besides Air Force cadets that we can study that have a lot of the variables set? And I'll show you why that's important in a moment. A lot of variables set so we can begin to look at the diversity or the variability that we're actually interested in. Air Force cadets are great for that. Okay, and here's my deal, the people, right? I really have spent my career looking at the people. I never thought I'd spent my career looking at the guts of people, but I really have been focused on this quite a bit. And so two projects I want to highlight today, and the one I'm going to talk about most is the Veteran Microbiome Project. In the interim, we also did receive a grant, and this is the lovely consort diagram, from the Department of Veterans Affairs to look at a biological intervention, L-reuteri, for treating co-occurring mild TBI and PTSD. So this idea of looking at inflammation and inflammation underlying both physical and mental health related conditions, I think also a lovely way for us to continue to break down the barriers between physical and mental health. I know we still think about those as being very, very different, but the reality is probably many of the underlying mechanisms that drive symptoms associated with physical and mental health are similar, and these often travel together. So I'm not going to talk as much about the SPIRE, we call it the SPIRE grant today, but I'm going to talk more about the Veteran Microbiome Project. The other thing we've really encountered is how do you socialize the idea that the microbiome could be important for mental health. And I'll say one of the first grants we put in, a previous version of the SPIRE grant, I think it was a panel of GI docs and GI researchers who were like, why the heck are we getting a grant on mental health? You're looking at the gut microbiome. So I really appreciate folks who are taking the time to highlight models like this one. This is a recent article that just came out that does a very nice job of beginning to highlight how the microbiome, the gut microbiome, could affect both peripherally and centrally to impact mental health and I would say physical health if I'm talking about traumatic brain injury, which I'm talking about a lot of the time. If you haven't checked out this article, take a picture. This is actually a really, really nice article, and I know I will be including it in my next grant because it's very easy for people to read and understand. So one of the early studies we did was a collaboration with folks from South Africa, and what we began to do is look at the relative abundance of specific microbiota. And what we found here was we could identify the relative abundance of key microbiomes, and that they actually did seem to parse out differences between those who had PTSD and those who didn't. I should say I don't think this is exactly the right model that we should be going for. And I should have said previously, and it's probably obvious to many of you, when you're looking at a stress diathesis model, when you're looking at variables or vulnerabilities in human beings, we would love to be able to say that mental health is a genetic related disorder. The work towards identifying genes that contribute to mental health conditions has been very complicated. And the entire field in terms of mental health is moving to kind of polygenic models because it's not going to be one gene, right? And I would say the same thing for looking at the microbiome, right? It's not going to be one thing. It's going to be combinations of things in the complex system of the human and their environment. But what we did also find in the study, interestingly, is not only was PTSD associated with these microbes, but individuals' self-reported score of the childhood trauma questionnaire, which is individuals saying, hey, I experienced this kind of trauma, was also associated with this, suggesting that these individuals had been carrying around this vulnerability for quite some time, and then they were exposed to trauma, and that trauma exposure actually is what facilitated or resulted in the development of PTSD. So back to, I think I love the earlier talk about how do we identify perfect or good periods around development where the differences really matter. And it may be that some of the differences in folks related to these mental health conditions happened long, long before people join the military, long, long before people are adults and have the onset of these conditions. If you think about the onset of most severe psychiatric symptoms and that's not PTSD because that takes a trauma, is around like late adolescence, early adulthood, but this could be set up way before. It also does support the old friends hypothesis that we are having less exposure to these old friends, thereby creating less microbial diversity and vulnerability to mental health conditions. Do want to highlight, some of you have heard about this work before. This is, so far, if there's funders in the room, this is a pet project. All this work has been done because we can't help ourselves. But this is modeled, although not half as fancy, with a $0 budget, after the Human Microbiome Project. This is the Veteran Microbiome Study. We have veterans ages 18 to 89. All you have to do is be able to sign a consent and give us your samples. We're taking oral, skin, and stool samples. To date, this isn't even our total sample. Oh, I should go back. The other thing we're doing that's a little bit different is we're providing a complete characterization of their psychiatric history, and we also have a complete characterization of their health history based on their electronic medical records from the VA. So we have, and if you don't know, and I'm happy to talk about the strengths and weaknesses of the VA, but actually the VA is a great place to get health care. And we follow people for a long time. And so we have people's onset of health and mental health conditions and can follow them longitudinally over time to see what happens with them, which I think is an amazing resource for this kind of research. So we have very well characterized samples. And then we also, I want to highlight, this is a riff on a previous talk that I've given. If you're interested in studying humans, don't develop your own measures. Psychometrically sound measures already exist. These are some examples. This is the Insomnia Severity Index, and this is the OQ45, which is a measure of psychological distress. But these are examples of some of the measures we have on all the folks that are in the Veteran Microbiome Study. So far, I'm going to actually show you data from 188. We have more now. Kelly, we have over 250 in the PET project. And so this is a little bit different if you're familiar with the sample of the Human Microbiome Project. Our mean age is 48, but our range is ages 24 to 77. It's mostly men. We have huge variation in employment status, student status. 9% of our sample was currently homeless. A larger part of our sample had a history of homelessness. You can see lifetime homelessness as 24%. We also have a huge range of service era and a huge range in terms of deployments. So this is 0 to 15 deployments overseas in different environments. Also combat tours. Andy provided me with this nice number. This represents 1,246 years of military service, or 6.6 years per person. So this is a very different sample than the very healthy, mostly young samples that have previously been collected. And I believe it's this kind of sampling with this kind of population that's going to help us figure out mental health conditions and other physical conditions. We're not going to figure it out if we have super healthy people that have no problems, right? That's not going to get us where we need to be. Okay, the other things I wanted to show you, just this is some of the data from the measures. This is already a measure I told you about. These measures let you kind of identify a severity of clinical syndromes or symptoms. So 11% of our sample had severe clinical insomnia, 23% had moderate clinical insomnia. You can see the distress measures. Over half, about half have significant clinical distress at the time that the measures were taken. Again, if you collect these standardized measures and you collect them over time, you can characterize not only symptoms, but how people are feeling and how they're reporting that they're feeling. So we also have collected some of these same samples, these same measures with the cadets. And so this is some of the data from them. Just to show you, so ISI scores, right? We have cadets all saying, we're all good. We have veterans saying, actually, we're not that great, right? And we also have veterans on the distress cell all over the map. And cadets also saying, hey, we're pretty good, although there's a few outliers, a little bit distressed. This does highlight, though, that our standardized measures are good if people are willing to answer them. Okay, so we do need objective ways to measure distress, measure some of these things. Actigraphy and the Trier would be two different ways to get at some of this level of distress that are more objective rather than subjective. But I think important to kind of look at how we use these measures and how could we collect this data over time. And then also what we've really been working on is with different samples collecting the same data so that we can start to talk about between samples. We've also been developing in the past 2 years our own pipeline and capability to run our own microbiome data. So here you can see, here's our skin, a stool, and oral sample, and it's looking pretty good. It looks like we're able to do this, which is rocks. I gotta say, I never thought I would be presenting data like this, but I'm so psyched that we can be able to do this. And then here just is the 20,000 foot level look. Here you see the Human Microbiome Project data, the Veteran Microbiome Project, and the Cadet Microbiome Project data. Again, 3 very, very different samples from 20,000 feet have a lot of overlap. That's the only thing I'm trying to show you on this slide, is that if you are looking at this very big picture or looking for easy answers, you are not going to find it, right? This is going to be a much more complex interactional problem for us to figure out. That being said, we did want to pull out a few highlights for you to look at. And so you can see here again, we've pulled out specific microbiomes that we're interested in and started to look at what do we see in these different samples. So here you have, Akkermansia is a microbiome that we identified previously in the study that I'd already talked about with PTSD. But you see in the cadet study, you've got a little bit of diversity. You've got more diversity in the human microbiome, and in the veteran, you've got a lot more. With this idea that like more is better here. And what we were able to show, and this is just the first time this is being shown, is that veterans who had more of this had no reportable diagnosis on the SCID. Right? And the SCID is a structured interview for any mental health conditions. So we're pulling back a little bit and saying, hey, is there a relationship between this one thing and any mental health condition? And the other thing it makes me think about is for the cadets that are low, right, surprisingly low on this, a number of the cadets, are these individuals at risk? Are they like the childhood trauma questionnaire that we showed before with the South African sample? Does this suggest some level of risk that we should be tracking over time for this cohort? Now again, I don't think it's going to be one thing. It's going to be combinations of things and events over time. Here's another example. Here, less is better. More has been associated with a lot of inflammatory diseases like rheumatoid arthritis. And again, we have it split out by SCID, reportable, no reportable SCID diagnosis, reportable SCID diagnosis. And how do we begin to take these broad categories of mental health and these specific pieces of data and put them together in models that can help us predict and prevent mental illness as we go? And then how can we use the built environment to either introduce or take away strategies to be exposed to things we want to be exposed to and not be exposed to things we don't want to be exposed to. I should also highlight that, obviously all these folks have been in the military because they're veterans, so they do have periods of being very intensely together in small groups. And so this may be an interesting cohort, like students in dorm rooms, other things, we think about families a lot, but there's a lot of other groups where people are really close together, where we have environments to manipulate over time. I'm not advocating for manipulating in any way today, but I think over time, thinking about how to do so would be useful. The other thing I just want to highlight, we named ourselves. I realized that actually people just name themselves, and we gave ourselves a name, and the name is the Military and Veteran Microbiome Consortium for Research and Education. We're really trying to work together to identify ways that will really help veterans and their families, but really the civilians on whole too. And we really want to thank Paula, because she thought this work is not that weird and maybe it's going to be helpful, and she's been a big support of us, so thank you, Paula. And I thank you all for letting a psychologist talk to you about the microbiome today, so thank you.
Time for questions for Dr. Brenner.
Nope. Okay.
Oh, we have one. Lisa.
All right. I should also say, we are on social media, so please follow us, and I'm on Twitter, and I'm a big fan, so please reach out.
Yeah, so I've done some recent studies with some the effects of steroid hormones on the microbiome.
Yep.
And when you have childhood trauma,
yes,
you're going to have high levels of glucocorticoids,
yes,
all through life, which could affect all sorts of
absolutely
bodily stuff. Have you looked at that, or have there been any studies about glucocorticoids per se?
So So there is some really interesting work around glucosteroids in terms of PTSD, and if you can, have you seen this where if you could administer them very early on, they look like they are protective in terms of developing PTSD? And so again, these ideas of like childhood, adult, shifting things that we don't really understand, both in terms of the host, but also in terms of the microbiome. And you know, as all of you begin to, if you are ever interested in the health-related stuff, you'll look at one article and one thing seems good, and you look at the next article and one thing seems bad. And I think it's really going to be about interactions over time and environments together. Thank you.